Good morning, everyone. We were discussing about the biopesticide formulations in our previous session, and we were also discussing about the mechanism of action of the biopesticide as against the pest or the attack of the pest. Now, in this session, we will try to understand the process of bio leaching. Essentially, it is a microbial process of metal recovery. As a consequence of technological and industrial development, so the many industrial sites which are contaminated with the heavy metals as well as organic compounds, now they are recognized to be anthropogenic and toxic to many kinds of living organisms and particularly human beings. So this is the outcome of the technological and industrial development, so that the industrial sites are contaminated with heavy metals and organic compounds, and these are referred to as anthropogenic, and they are toxic to many living organisms including the humankind. So therefore there should be a consensus or an obligation between the industry and the government to implement the concepts of structured environmental management system in a more effective way to reduce the environmental burden of toxic substances. So, the application of environmental biotechnology has improved the cost and efficiency when microorganisms are exploited for the sole purpose of reducing the environmental burden of toxic substances. So, there should be an understanding between the industry and the government to strictly implement the concepts of structured environmental management system with the sole objective of reducing the environmental burden of toxic substances. If we just recollect what we have studied in our previous sessions, there are various bioprocessing technologies to reduce the toxicity of or affecting the environment. Number one is the bioremediation. Number two is biosorption. Number three is bioaccumulation. And number four is bioleaching. We know that bioremediation, it consists of the treatment of acid mine runoff or mine tailings to reduce the contaminants affecting the natural bodies. Whereas the biosorption and bioaccumulation, it readily activate on heavy metals to detoxify the environment where these metals are accumulated. So it is essentially activating on heavy metals and detoxifying these heavy metals in the environment wherever they are accumulated. So the application of microbial technology in the mining industry has shown a good progressive development in metal extraction way back in 20th century itself. So this process is commonly referred to as bio leaching. So in technical way to define or to understand what is the bioleaching process, in general, bioleaching is a process which is described as the dissolution of metals from their mineral sources by certain naturally occurring microorganisms, or the use of microorganisms to transform elements so that the elements can be extracted. So this is a usually found definition to understand the process of bioleaching. That means it is described as the dissolution of metals from their mineral sources by certain naturally occurring microorganisms, and or the use of microorganisms to transform elements so that the elements can be easily extracted. Right. Now, if we just look at the principles of microbial metal leaching, the microbial metal extraction process. So these processes are usually more eco-friendly when compared to the physico-chemical processes. So this is most important. Eco-friendly refers to the good deed of environment, so that these methods are eco-friendly when compared to the physico-chemical process, and they do not, that is the microbial metal extraction process or the bioleaching what we are referring to, they do not use a large amount of energy, and they do not produce sulfur dioxide or any other harmful gases as compared to the physico-chemical process. See what happens in physico-chemical process. There is a step for roasting, there is a step for smelting, and these steps they require large amount of energy, whereas the microbial metal leaching process do not use large amount of energy as compared to the roasting or smelting process that are the part of physicochemical process of metal extraction. So the microbial metal extraction process do not produce harmful gases such as sulfur dioxide, and hence it is eco-friendly. As such, this process can be considered to be more compatible with anti-pollution laws of any country that is or that exists. Is that clear? Now, the principles of microbial metal leaching refers to the mineralogic effects of bacteria and fungi on minerals, and they are mainly based on three principles, right? So the principles of microbial metal leaching with respect to mineralogic effects of bacteria and fungi on minerals, they are mainly based on three principles. Number one, acidolysis. Number two, complexolysis, and number three, redoxolysis. Okay, number one, acidolysis, number two, complexolysis, and number three, redoxolysis. If we just try to understand a little more, microorganisms they are capable enough to mobilize metals through three different processes, right? So the microorganisms, this is essentially we are trying to understand the mechanism. So microorganisms are capable enough or they are able to mobilize metals through the three important processes. That is number one, formation of organic and inorganic acids. So it is referred to as proton formation, right? Number one, formation of organic and inorganic acids we refer to as proton formation. And number two, the second process, excretion of complexing agents, that is ligand formation. Number two is excretion of complexing agents, that is ligand formation. Number three, oxidation and reduction reactions. Right. So this is essentially acidolysis, complexolysis, and redoxolysis. So number one, formation of organic and inorganic acids, that is proton formation. Number two, excretion of complexing agents, that is ligand formation. And number three, oxidation and reduction reactions. So generally, sulfuric acid is the main inorganic acid that is formed in the leaching environment wherever you find the leaching happens. So it is the sulfuric acid is the main inorganic acid that is formed in the leaching environment. So it is formed by sulfur oxidizing microorganisms such as thiobacilli, and also a series of organic acids are formed by bacterial and fungal metabolism, resulting in organic acidolysis and complex and chelation or chelate formation, right? So the proton induced and ligand induced mineral solubilization occurs simultaneously in the presence of ligands and acidic conditions. Okay, now if we just try to understand the mechanism in a defined way, the key aspects of the leaching have been integrated and a mechanism has been developed. So the mechanism is as follows. It is a very simple way to understand the mechanism what we have studied so far. Number one, cells have to be attached. So it is like a flow chart, and you can visualize how the mechanism happens. The cells have to be attached to the minerals and should be in physical contact with the surface. And number two, the cells form and excrete exopolymers, right? So this is we are speaking about the mechanism in a flow chart way. Number one, the cells have to be attached to the minerals and they have to be or should be in physical contact with the surface. And number two, the cells form and excrete the exopolymers. And number three, these exopolymeric cell envelopes, they contain ferric iron compounds, and these are complexed to glucuronic acid residues. Now, these are regarded as part of primary attack. So, this is the most important step in the mechanism of bioleaching. And number four, the thiosulfate is formed as intermediate during the oxidation of sulfur compounds. And number five, sulfur or polythionate granules are formed in the periplasmic space or in the cell envelope. So essentially, the mechanism of bioleaching gives us an idea how the microbial cell is involved in leaching or extraction of the metals from the contaminated sites. So this is about the process of bioleaching, and now we will move on to a new concept under DNA cloning and sequencing unit 2, that is about transgenic plants. And the transgenic plants, we know that the humankind has genetically manipulated plant crops for many years through conventional breeding. And until recently, breeding was the only way to introduce the phenotypic characteristics of interest, which are determined by the genes of an individual plant or species. Now, the desired phenotypic traits, they are transferred to the progeny through breeding and selection. So we are trying to understand how the transgenesis happened, and with the preamble of the conventional plant breeding and approaching to the transgenesis or the method of transgenesis. However, such conventional methods of genetic manipulation have some limitations. And these limitations are number one, sexual barrier between the species. So, this is the most important limitation for conventional methods of gene or genetic manipulation. Number two, phylogenetic isolation barriers between and within genetic groups. And number three, a reduced gene pool. And number four, linkage drag. So, number one, sexual barrier between the species. Number two, phylogenetic isolation barriers between and within genetic groups. And number three, a reduced gene pool and linkage drag. So, all of these drawbacks are in addition to the lengthy time usually required for desirable traits to be transferred. Is that clear? And most recently, the molecular biology, tissue culture, and the gene transfer techniques, they have been combined together into a powerful tool. See, all these techniques, they do not or they are not used in separately, but they are combined. When they are combined, they become a powerful tool for the introduction of new traits into a particular plant. So what happens as a result, the researchers or the scientists now, they can introduce genes from animals, microorganisms, or any other different plant species into the genome of the recipient plant in a very controlled manner and independent way of fertilization. So it is totally independent of fertilization. Fertilization is essentially not required with the tool, the powerful tool that they have from molecular biology, tissue culture, and the gene transfer techniques. Now it is possible to introduce genes from animals, microorganisms, or different plant species into the genome of the recipient species. When we are speaking about plant transgenesis, it is about the plant as recipient. So this can be done in a very controlled manner, and it is an independent way of fertilization. So it is not dependent on fertilization at all. So hence the sexual barrier between the species are eliminated. So when it is independent of fertilization, definitely sexual barriers between the species are eliminated, as well as the barriers between organisms of the eubacteria, archaea, and eukarya phylogenetic domains. So absolutely there is no sexual barriers between the species. It is completely eliminated because the entire process is independent of fertilization. Right. So the plant genetic transformation, how to understand the plant genetic transformation, or how do you define the plant genetic transformation? The plant genetic transformation can be defined as the controlled introduction. It is not just introduction of something, but it is the process is very much controlled. That is controlled introduction of what we are trying to introduce, a specific segment of DNA into a recipient genome, that is plant genome, because we are speaking about or discussing about plant transgenesis. So the controlled introduction of specific segments of DNA into a recipient plant genome, excluding an introduction by fertilization or hybridization. Right. So this is how the plant genetic transformation is defined. And other non-sexual gene transfer techniques exist for plants, such as somatic hybridization, wherein the two individual protoplasts, they are fused, mixing the nuclear and cytoplasmic genomes. So apart from this, we have the another non-sexual transfer method, that is hybridization, which occurs through cytoplasmic fusion. So in somatic hybridization, where in which the two individual protoplasts are fused, mixing the nuclear and cytoplasmic genomes, and in hybridization, which occurs through cytoplasmic fusion. Right. So genetic transformation in plants, it was only made possible by the development of plant tissue culture techniques. So it is totally dependent on the plant tissue culture technique, and it was possible, and it was made possible, and it was made real, or it was in reality only with the development of plant tissue culture technique. So for the transformation process to be effective, the foreign DNA must first be introduced into a plant cell or plant tissue. Then a transgenic plant is then generated from the transformed cell. So essentially, this is what the transgenic plant generation process is. The transformation process, it can be effective only when the foreign DNA is introduced into a plant cell or the plant tissue, and then the transgenic plant is then generated from the transformed cell. So this process of transformation is enabled by the totipotency phenomenon, which is that is the ability of an individual plant cell to develop into entire new plants under favorable conditions. So usually we define totipotency as development of entire new plant, but we forget to mention about the favorable conditions and in the presence of growth regulators and nutrients. So this is most important when we try to understand what the totipotency is. So the genetic transformation of or the process of transformation is enabled by the totipotency phenomenon, which is the ability of individual plant cells to develop into entire new plants under favorable conditions and in the presence of growth regulators and nutrients. Right. So if we just look at the illustration over here, before we try to understand about the genetic transformation, we need to understand about the gene organization and the process of gene expression, which is very much essential for genetic engineering processes, right? So if you just carefully look at this image, so the entire concept of gene organization and gene expression is given in a nutshell. So initially the RNA polymerase binds to the promoter region and reads the DNA from the 5 prime UTR to the 3 prime UTR untranslated regions. So a pre-mRNA strand is synthesized and it receives a cap, that is which consisting of the modified nucleotide 7-methyl guanosine triphosphate, or that is M7G triple PNP, to the 5 prime end, and a repeated adenine sequence, that is a poly A tail, to the 3 prime end. So I'll repeat once again. So the process is like initially the RNA polymerase, it binds to the promoter region and reads the DNA from the 5 prime UTR to the 3 prime UTR, and then a pre-mRNA strand is synthesized and it receives a cap, that is it consists of a modified nucleotide, that is 7-methyl guanosine triphosphate, to the 5 prime end, and a repeated adenine sequence, that is AAA, that we refer to as poly A tail, to the 3 prime end. Then what happens is the pre-mRNA is spliced to remove the introns and transported from the nucleus to the cytoplasm. What happens in the cytoplasm? So in the cytoplasm, the mature mRNA is coupled to ribosome subunits, and once it is bound to the ribosome, the mRNA receives its amino acid tRNA, which contains amino acids specifically matched to each anticodon, and the polypeptide chain is synthesized. That is, you end up with the product, the proteins. So this is essentially to understand the gene organization and gene expression pattern, to thoroughly understand the genetic engineering processes, right? So if we just move on further in understanding the manipulation of the nucleic acids, there are several steps which are involved in the process of creating a transgenic plants, and these processes are: number one, gene identification, then isolation and sequencing. So number one is gene identification. These are the steps that are involved in the process of creating a transgenic plant. So how you start out with creating a transgenic plant? So how the transgenic plants are created? So they include number one, gene identification, number two, isolation, and number three, sequencing. And another critical phase is cloning engineering. So this cloning engineering means it involves, or which involves, the construction of the insert which contains known coding region and its promoter and terminator regions. So cloning engineering essentially involves the construction of the insert which contains the known coding region and its promoter and terminator regions. So it is about the gene identification, isolation, and sequencing. Now, to isolate the genes of interest, a genomic library must be created and probed. So, the process of transgenesis is quite, it has several steps. And to begin with, to isolate the gene of interest, how you go for isolation of the gene of interest? The gene of interest could be for any trait, right? Resistance against the pest, or resistance against the drought conditions, or for better yield, so something like this. So to isolate gene of interest, a genomic library must be created and probed, and if the identified gene is expressed in a sexually compatible species when compared to conventional breeding, and then conventional breeding can be employed. So there should be compatibility. The identified gene should be capable enough to express in sexually compatible species, and then you can carry on with the conventional breeding. So you can have a combination of genetic engineering process along with conventional breeding. And all of these processes collectively, they are referred to as the recombinant DNA technology. Right. Now, some of these technologies, what we refer to as recombinant DNA technology, the sub technologies, what you to make you understand in a better way, they are the involvement of restriction enzymes, and you see cloning vectors. There are DNA ligases and bacterial transformation methods, and there are methods to distinguish between the transformed cells from those that were not transformed. So essentially, we are trying to understand each one of them so that the creation of transgenic plant will be understood in a better way. So restriction enzymes, we all know that they are also called as restriction endonucleases, and they recognize a specific sequence of nucleotides in double-stranded DNA, and they cut the DNA at a specific location. And these restriction enzymes are indispensable to the isolation of genes and the construction of the cloned DNA molecules. Generally, these are the cleavage sites, or the sites where the restriction enzymes act upon. They are symmetrically positioned, or we call it as the palindromic sequences. So once the genes are or the identification process is completed, that is, the recognition sequence is completed, uh, the isolation of the DNA fragment which contains the genetic information of interest, it happens, and then it is inserted into a DNA molecule using a cloning vector. So once you have the genes identified, and then you have to insert the DNA, isolate the DNA, and then insert the DNA using the cloning vector, which is capable enough of copying and amplifying the genetic information. So I have a table over here where some of the restriction enzymes are listed along with the specificity, their specificity. We have the BAM H1 ECO R1 HIND3. So we have HA3, and the specific recognition sequences are given over here. It is only for the purpose of understanding the recognition sequence of each of the restriction enzymes. And this is not the entire list of the restriction enzymes. Now we have thousands of restriction enzymes available. So the primary cloning vectors which are used in plant transformations, they are derived from the bacterial plasmids. So the construction, that is genomic library constructions, they are constructed by cloning the entire genome of an organism. And how it happens: first a collection of all of a species, whatever the species you are intended to carry out uh, the identification and isolation. The DNA fragments is generated using restriction enzymes or the mechanical shearing, and these fragments, they are linked to a vector which is capable enough of receiving large lengthy inserts, and these could be the yeast artificial chromosomes, or the bacterial artificial chromosomes, abbreviated as the BACs and YACs, that is yeast and bacteria referred to for Y and B, so that a smaller number of clones are required to obtain a representative sample of the entire genome. And then the large inserts, they are also allowed for cloning of a complete gene in a single clone, or a small number of different clones, including its 5' and 3' flanking sequences. So essentially, this is about the process of transgenesis in a nutshell, and I hope you have understood, because these concepts you have been studying throughout your graduation program. So we will understand the fundamental principle of transgenesis with respect to plants and animals. So in the next session, we will understand the methodologies for the development of transgenic plants. So with this, I'll take your leave and I'll stop my discussion over here. Thank you very much for your kind listening. Thank you.